With histology of muscles. So the muscle tissue is mainly made up of cells known as myocytes. These are nothing but muscle cells or fibers. So the connective tissue forms the framework of the muscle and it provides the pathway for blood vessels, nerves to reach the muscle fibers. So the most common muscle in a limb is our skeletal muscle, which is striated and it's voluntary, that is under our control, and it is supplied by our somatic motor nerves. And most common muscle in a viscera is a smooth muscle. It is non-striated, that is it's not having any stripes like structure, and it is involuntary, that is not under our control. So sometimes the smooth muscle may also be voluntary, like in the cases of passing out urine, etc. So smooth muscle is supplied mainly by the autonomic nervous system. And the most common muscle in the heart, we know that is cardiac muscle. It is striated and it is also involuntary because heart doesn't pump according to our wish. It's involuntary, not under the will, and it is supplied by the autonomic nerves. So the involuntary muscles are smooth muscle and cardiac muscle, but the skeletal muscle is voluntary, which is under our control. And skeletal muscle supplied by somatic motor neurons, smooth muscle by autonomic, and also cardiac by autonomic. So all the involuntary are supplied by the autonomic nervous system, but the voluntary one is supplied by the somatic motor nerves. So sometimes the skeletal muscle becomes involuntary in cases like sneezing, coughing, respiration, so that will be in our control sometimes, but smooth muscle will become voluntary in the case of passing the urine. So as you know, skeletal muscle should be voluntary, that is always in our control, but sometimes it is not under our control. In case of respiration, it is not under control, coughing, sneezing, etc. And smooth muscles should be always involuntary, but sometimes they are voluntary, like passing urine. So skeletal muscles are long cylindrical fibers having greater than 30 cm in length, and they are having the multiple nuclei, and they are peripherally located under the sarcolemma, that is nothing but the plasma membrane. So the sarcoplasm is nothing but cytoplasm having the longitudinal fibrils known as myofibrils. So the myofibrils, they are present in groups and they are nothing but known as fields of cornea. And the dark bands, so our skeletal fiber, myofibrils have dark bands and light bands. So the dark bands are the A bands and the light bands are the I bands. So what are these dark bands and light bands? Let us understand by drawing a structure over here. So these are the light bands. As you can see, I am drawing in lighter structure. So in middle we have a dark band like this, dark bands. So these light bands together form a structure like this, and they form a Z line over here, and in middle we have one M line. So the unit between two Z lines, that is two thin bands, is nothing but known as sarcomere, and the thick bands are nothing but known as myosin, thin bands are known as actin. So let us see here. So the dark bands are the A bands and the light bands are the I bands. So A means I means isotropic, A means anisotropic. So A bands are having middle H band, A bands, that is our dark bands are having in middle. So these are the dark bands. So in middle they are having one line, that is nothing but our H line. And so light bands are I bands, their middle having a Z band. So these are I bands, right? These are light bands which are forming the Z band, right? Dark bands are forming the H line. So remember that. And next, myofibril between two Z bands is known as sarcomere, as we said. And the glycogen provide the energy for the contraction of the muscle. And the fasciculae are nothing but the group of muscle fibers. So first let us come from inside to outside. So inside we have one myofibril. So many myofibrils together form a fasciculae, and the fasciculae is nothing but the group of muscle fibers. And the connective tissue supporting the myofibrils and also unite them. So the individuals are covered by endomysium. That is, first let us say, for example, this is a myofibril. So this myofibril is covered by a endomysium because it's innermost structure, so it is covered by endomysium. So the fasciculae, as I said, group of muscle fibers. So for example, this all are myofibers. This group is covered by perimysium. So innermost endomysium, this is perimysium, and the entire muscle. Now this group, even one more group, for example this pen, one more group. So these two groups together are covered by and entire muscle, that is outer, it is covered by epimysium. So the muscle and the tendon together have endomysium, perimysium, epimysium, which are nothing but continuing with the tendon. So what is tendon? Tendon is nothing but having the collagen fibers in the form of bundles, and they are united by a areolar tissue which contains the fibroblasts, and the longitudinal section shows the tendon along with the fibroblasts are elongated, and in the transverse sections, if we see, the fibroblast is having the stellate cells. So innervation. So how the skeletal muscles are innervated? Mainly they are innervated by nerves, and blood vessels enter by the neurovascular hilers near to the origin of the muscle. So the nerve break and form the branches, and they run into the connective tissue of both perimysium and endomysium and reach each muscle fiber. So the skeletal muscles are supplied mainly by axons of large neurons in the anterior gray column of our spinal cord, of nuclei of the, and also from the nuclei of the brainstem, respective nuclei. And the alpha efferents are mainly myelinated, and they increase the they're having more diameter, and they may supply many muscle fibers. So the junction between the muscle fiber and a nerve terminal is having a motor end plate, and every muscle also receive the gamma efferents from the ventral gray column of the spinal cord, and they supply the muscle spindles. So the special muscle fiber is called as intrafossil fibers. So the structure, let us see the myofibril structure. So outer we have the basement membrane or extra lamina, and it continues to form sarcolamella, and inside we know we have the endomysium, which is lining a myofibril. So between the myofibrils we are having a sarcoplasmic reticulum, and also having elongated sarcosomes. So sarcosomes are nothing but mitochondria, etc., other organelles. So the structure is having a fine myofibrils filaments. So let us see the fine myofilament, like this is one myofilament, and this is further divided into actin and myosin, that is the thin band and the thick band. So if you remember, the thin band is always related to the Z line, that is actin filaments meet here, and thick band is forming H line, if you remember, and also thick band is always related with the M band, that is interconnecting on between the myosin. So actin is thin, myosin is thick, and the H band is also a part of A band, that is a thick band, right, as we discuss here. So thick band is related with two lines. What is those? H band and M band, both are related with the thick, and thin is only related with the Z line. So remember, thin only Z line. The thick band is related with both H and M. You can remember like H and M. So the H and M are related with the thick band, and the H band also a part of a actin filaments, and they don't extend, and the actin filaments don't extend in the case of H band. Next, let us check out our contraction of myofibril. So contraction of myofibril, so whenever this light bands will be there only, thick bands move towards and they just contract. So whenever they contract, they shorten the fibril, that is both actin and myosin, and the sliding of actin is more into the myosin, that is the light band is more sliding towards the myosin, and the width of the I decreases, that is width of our I band decreases, but the A band is same, that is isotropic band is decreasing and anisotropic band is same. If you remember, isotropic band means light band, so width of light band is decreasing because actin is more moving towards myosin, means A is more sliding, so thin band is decreasing, width of the thin band is decreasing. So the actin filament is actually made up of G actin, which chains are twisted around to form a chain which is known as F actin. If G actin forms, that is G actin is like one round molecule, and all those together they twist, there are two chains, so they twist like this. If they twist and form a chain, then we call it as F actin. So the actin filament has both head and tail. So the head extend up to the A band, that is the thick band, and the tail anchors the Z line by alpha actinin protein. And our actin filament is having two proteins, that is tropomyosin and troponin. So the tropomyosin is winding around the actin, and the troponin represents the sites where the myosin can bind to the actin. So troponin produces a surface where a myosin can bind, and tropomyosin is winding around the actin filament itself. So myosin filament is also having both head and tail. So the head is outward projection and it drags the actin towards it, and the So first it breaks the bonds and again start dragging. So this breaking and adding bonds is mainly dependent by our ATP, and this leads to the contraction of the muscle. And the tail is mainly attached to the M line and it is coiled over each other. So the myosin and actin together form the movement or the contraction of the muscle. So this process requires necessary proteins for support, and also energy of binding and releasing is mainly done by the hydrolysis of ATP. So now whenever the hydrolysis of ATP takes place, then the ADP is formed, then when the head is released. So when head want to bind, then the ATP is formed, and when head want to release from the actin filament, then ADP is formed. So other proteins of our filaments, not only our myofilaments have these proteins like troponin, tropomyosin, we also have some other proteins like actinin, which is present in the Z disc, which bind the tail to the actin to the disc, and the myomycin in the M disc, which binds the tail of myosin to the disc, and titin, which links the head of the myosin to the Z band, and the desmin is present in the intermediate filaments of the cytoskeleton, which links the myofibrils to others and also to the cell membrane. So the cytoskeleton linked to the actin, and it is associated with the external lamina and also connected with the connective tissue in the muscle fiber, and sometimes there are genetic defects in it which lead to abnormal muscle formation. This is known as dystrophy. So contraction of muscle mainly dependent on the calcium. In resting, it is bound to the sarcoplasmic reticulum, but whenever it is active, it depolarizes into centrotubule, that is calcium go to the myofibril and contraction takes place. Otherwise, it is bound to the sarcoplasmic reticulum. So let us check out sarcoplasmic reticulum. It is between the myofibrils, and there are tubules present in a sarcoplasmic reticulum which are forming rings. So between A and I, there are muscles triad, that is upper, middle and lower. The middle one is centrotubule, it is also known as T system. Upper and lower together form a network by small tubules which are mainly present opposite to both A and I band, and they are known as closed system of tubes. This is sarcoplasmic reticulum. So the skeletal muscle is having white and red rich fibers. So the white is contracting fast, and it is also known as fast twist or type 2, and red type 2 cells, and red is known as slow twitch or type 1 cells. It is also known as postural muscle. So red is mainly due to that red color is mainly due to myoglobin. It is also present in white but in low quantities, so we can't it doesn't appear as red. But in red, as it is more, it appears in red color, and it is narrow and it has less amount of mitochondria. More amount of mitochondria and also more amount of sarcoplasmic reticulum, and the capillaries are also more. In white, the type 2 cells are further divided into 2A and 2B, which differ in chemicals and also enzymes. So the skeletal muscle is mainly having rich blood supply, which blood vessels, and which enter at the neurovascular hilus, and the plexus is mainly present in the epimysium and perimysium, and also there is more amount of red muscle. So the epimysium and perimysium also have the lymphatic plexus. So now let us check out the cardiac muscle. So the cardiac muscle have elongated fibers, and they branch and anastomose, and they are having multinucleated syncytium in the each fiber. Cardiac myocytes have nucleus, they are having their own nucleus, and also which is centrally placed, but in the case of skeletal muscle, it is peripherally placed. And increase of sarcoplasm, mitochondria close to the red type of, it is mainly related to the red type of skeletal muscle, if you remember, as we discussed this red type. And there is more amount of myoglobin, glycogen, and capillaries are present around the fibers, and centrotubules of the system lie opposite to the Z band, and wider tubules are present in sarcoplasmic reticulum. It is replaced by dyads with type 1 of tube and 1 tube of sarcoplasmic reticulum, and they are having transverse dark lines, that is intercalated discs, which are mainly opposite to the I bands, and this intercalated disc is formed by the cell membrane of adjoining myocytes, and the myocytes are connected to the desmosomes mainly, which link the intermediate filaments in the cytoskeleton of adjacent cells, and they are also having gap junctions, which are electrical connections which are forming the physiological syncytium, and they are also having the tight junctions. So the actin filaments end in relation to the tight junction. So the cardiac muscles are mainly involuntary, their supply is by autonomic nervous system, and then nerve ends mainly in the myocytes but not in the motor end plate as we seen in skeletal muscle, and these are isolated myocytes contract with the rhythm, and it is pacemaker pacemaker which is present in our SA node. Now let us start with our smooth muscle. So the smooth muscle is non-striated, doesn't have any striations, it is involuntary and it is a plain muscle and having the myocytes and also having the taper ends and the broad center. So the nucleus is oval, elongated in the center, and it is having no transverse section, only longitudinal striations are present and aggregates to form the bundles, fasciculi layers by which surrounds the collagen, reticular, elastic fibers, and the distribution, it is most common in the wall of the hollow viscera, especially in our stomach, intestine, urinary bladder, which hold the myocytes together, and in narrow tubes like arteries, veins, bronchi, uterus, and also in different uterus, uterine tubes, ducts of glands, etc. And in it is helpful in constriction and dilation of pupil muscles, and also present in orbit, that is in our orbitalis, upper eyelid, that is Muller's muscle, and it is present in prostate, in skin of scrotum, dartos muscle, hair follicles, bundles as erector pillae. And the arrangement you can see in gut, it is arranged mainly as two layers, that is inner circular, outer longitudinal, which allows the peristalsis and also propulsion of food. And in ureter it is arranged as a reverse arrangement, inner longitudinal and outer circular one, and sometimes it may also have three layers, that is inner and outer longitudinal with a circular layer in between. So in urinary bladder, uterus, it is having many layers, and in bile duct there is thick circular muscle surrounded by the segment of tube forming the sphincter. And the narrow bands are present in the skin. It is mainly innervated by autonomic nervous system, by sympathetic and both parasympathetic. Sympathetic and parasympathetic have different function, like in iris, if you take, if parasympathetic is innervating, then it constricts the pupil, and in sympathetic it dilates the pupil. So let us come to the blood vessels and lymphatics. These are present, but the blood vessels are less when compared with the skeletal muscle. And the smooth muscle is mainly bounded by plasma membrane. Outside it is having external lamina which is associated with connective tissue, and there are numerous invaginations known as caveolae which resemble the endocytic vesicles near the surface of the myocyte. So myosin and actin together only if the light chain is phosphorylated, in which leads to contraction in the case of smooth muscle, and the actin is different, and there is no troponin in a smooth muscle, and the smooth muscle need less amount of ATP for contraction, and calcium can flow by caveolae. Actin and myosin can form bundles at the ends on cell membrane called anchoring point, that is in contraction, which come nearer. And gap junctions are present, neurogenic, nutrient muscle contraction in iris, arteries, duct is different, and it is multi-unit. Neurogenic contraction takes place in a multi-unit muscle. And in unitary smooth muscle is a pacemaker in stomach, intestine, ureter, uterus, and smooth muscle resembles the fibroblast. Myocytes can produce collagen, elastic fibers, connective tissue matrix. Myoepithelial cells are contractile cells closely related to the secretory elements in some glands, and they squeeze the secretions. So myoepithelial cells are stellate, form the baskets around the echinae or maybe fusiform, and present in salivary glands, mammary, sweat glands, ectodermal in origin, and have the desmin. And myoepithelial cells are mainly innervated by the autonomic nervous system. So clinically, the skeletal muscle, when it undergoes hypertrophy, especially in case of exercise, cardiac muscle may undergo hypertrophy when there is increase in load to the chamber, example in the case of hypertension or any other reason. Smooth muscle undergo hypertrophy in the uterus, especially in pregnancy, and smooth muscle and cardiac muscle have less regenerative capacity, but skeletal muscle can't have the regenerative capacity, only their stellate cells can have persisting myoblasts. And excessive contraction of smooth muscle in the bronchi leads to asthma, and also colic pain, renal colic, biliary colic, etc. And the myofibroblasts proliferation in the tissue repair with the cirrhosis of liver, fibrosis of lung, and also causes atheromas of arteries. So absence of dystrophin is nothing but causing the Duchenne's muscular dystrophy. So all these are some of the important points from our histology of our muscle. So by this we complete the histology of muscle.